Any questions before we start? No? Okay. Where are we in the course? So today the third compact mode. The first compact mode we discussed knowledge of proteins and nucleic acids, how you can experimentally determine structures by X-ray diffraction, NMR, electron microscopy. Then there was a large chapter about databases, which is not that much theory, but more of applying these, knowing where to find information. And then last time, so we talked about this last time, and also about structure quality, what you have to look at, resolution, R factor, B factors, things like that. And then structure and functions and protein interactions. And actually this presentation was split into two. So last time, the first part was lecture 8, was about macromolecular interactions. And actually today we will continue with that. So we'll talk about actually drug targets. So this will be the first presentation. That's not that many slides that I will discuss. And then this is actually the large part of today's structure prediction. And then two rather small chapters about structural genomics and unstructured proteins. So for today, the major part will be about the structure prediction. That's for the lectures, for the presentations, and then I will also have a look at the end after I discuss these at the workshops. So I already published all the exercises. We go to the modules. So I will close all of these. So in fourth, you see that you have solutions 6 to 10. So this is actually for workshop 3. So these are all about molecular visualization, and you already know that you will get 2 molecular visualizations during the exercise and the exam. The workshops that I will focus on today then are workshop 4, workshop 5, so this is about Python scripting, 5 is about databases and tools, and then the last one here is about docking and homology modeling. I can already tell you that although I will talk today about docking and homology modeling, that you won't get such a task on the exam. Why? Because it's not feasible to do that in 3 hours. You would need at least half a day if you already know how to do this. So for you, I think in 3 hours that will be impossible to do. So you will have no docking or homology modeling to do during the practical part of the exam. Nonetheless, this is quite interesting. So for sure you have to look at the theory, but also it's nice just to have a look at the workshops. I thought, do I have a movie here? I have a recording of a movie here. Well, then I'll just talk you through it at the end of this video. And these are an example of what you can do actually. I also uploaded a YouTube video and supplemental information. Actually, a student of mine found this and sent this to me. Actually, a very good YouTube movie, let's say, summarizing large parts of the course. So if you want to quickly refresh your memory and you have some time, you can have a look also at this YouTube movie. Some of the tools that we use during the course, they generate PostScript files. On Windows, you might have a problem viewing these files. For example, I think I will show one example today of the last chapter. So I added this today. If you would want to look at these PostScript files, how you can install a program that's free to actually have a look at these PostScript files. If you won't have a program installed on your computer, then you can have a look here and install that. But that's supplemental. Okay, so let's start. Macromolecular interactions is what we talked about last time. And the last slide that I showed, summarizing this, is this one here. So actually summarizing all of the things that we know so far. And today I will also talk about docking here. So why is this important? Actually, if you want to, for example, have a look at drug targets, see how chemical molecules also, but also other molecules, small proteins, might interact. For that, you can do docking and ligand design. So drug discovery is actually multidisciplinary, where you can combine knowledge of chemistry, pharmacology, with then also contributions for biochemistry, and then actually looking in 3 dimensions, how, if you understand this, how it actually works, these interactions. That's actually what docking is about. One concept that is important to understand is the lock and key principle. So if you think about a door, if you want to unlock the door, of course, you need a good key to do that. This is actually more or less the same principle. And you have 2 aspects in this. You have a smaller molecule always and a larger molecule. Okay? So the smaller molecule is what we call the ligand. The larger molecule is what we call the receptor. It comes from ligand, from binding in Latin. And then here you see the second one is in what we call the target molecule, is typically the bigger one, the receptor. Of course, this doesn't mean it has to be a cell receptor or kind of protein like that. It's just the term we use to identify the larger molecule in the interaction. It can be one-on-one, but you can also have a complex, of course, of multiple copies of ligands, receptors, or even more than 2 molecules. That's, of course, also possible. And then, of course, you also have to understand the forces that are involved, and actually that's the same what we already know previously. For example, you can have electrostatic interactions, you can have positive residues on one molecule. If you would have a negative one, of course, that can form an interaction, but can also be hydrophobic. If you would have a protein normally that's hydrophobic, already told you that most cases that's on the inside. If it would be on the surface, that would just mean that that's actually a part of the surface that will interact with a similar molecule, same one. If it's a dimer, it could also be another one that forms a hydrophobic patch. And then, of course, that's again shielded from the solvents and could be an interaction surface. So different possible ways, of course, how these molecules can interact. And then in docking, actually, we try to understand how this works. There are tools that you can use to do this docking. We've used it in the past in this course, but for now, I will show you later on this evening. The easiest way for you to do that, if you would have to do that, is the automated docking is quite good already. So there are servers available, very good servers. If you just upload 2 PDB files, they will actually predict the most likely interfaces on those molecules that are able to interact. And then you will get certain clustering scores, but I'll talk about this later this evening. So you can combine 2 components, a search strategy and a scoring function, and you will see that coming back later this evening. If you want to divide it and have a look at different possible methods, we can split it up in rigid body docking methods and flexible docking methods. So the focus here is on the flexibility. So in this case here, both molecules, ligand and receptor, you say that they are not flexible. You try to look at the interaction. In the second case, of course, can be that your ligand or receptor might be a bit flexible. You can have loops that can bend, for example, so your molecule is not that rigid in this case. So that's another way of having a look at docking. Of course, this is computationally more extensive because you have more conformations to consider. And this is actually from the handbook, a scheme, how you can see different ways of doing this. So here you see searching function and scoring function also, and then you see a number of possible types of search algorithms, for example, that you can use, and here are some that you can also find here that you should know. Shape matching, so again, look and key principle, perhaps you know, you can have one key to unlock the door, but you can also have what they call is, how do they call it again in English? A skeleton key, a loper in Netherlands. Okay, so a loper, you can also unlock several doors. So it's a bit the same principle. For example, in drug targets, sometimes they just look at a certain molecule that can actually fit in to the receptor site. Okay, and then they might try to adapt that molecule, changing certain positions in that molecule, changing the atoms, and optimizing that. So for that, the principle there is the shape shifting. But you can have other fragment-based methods that you work with smaller fragments to look if they fit into, for let's say that you have the frame is okay to fit it in, and then you can again optimize that. So these are 4 different types of search algorithms, for example, that you can use in docking. Which tools are available to do docking? These are known, this is a publication already from quite some time ago, from 2006, it says here around 2005, 2006, where they had looked at how popular are certain tools. And I think it's obvious that one is more favorable, of more use than all the rest, and it is the Autodock. Maybe you already heard of it. So it's very popular, and the advantage of this tool is that it's open source. So you are able to just download, install it, and use it. And this is actually the tool that we used to try, and that I gave a demo in this course a couple of years ago. The problem with this is it's quite difficult to do that and to learn that. So during the workshop of 4 hours, if I would have a classroom of 10 students, only 1 student was able to follow until the end of the demonstration and be able to do the docking with the Autodock. So this is not that easy. So you actually have to spend, I think, a couple of days if you want to learn this, try it out, if you would have to do this. There are also other tools available. You see here Gold Flex, for example, also more than 10%, but these are commercial packages, so which you have to pay, of course. So this is described here, the Autodock. You actually have also a more recent version, is the Autodock Villa, and both actually are working a bit differently, but both can actually be downloaded from this website. But then, of course, it took quite some time to do this during the day course. And then I've actually found a paper here. So there are a number of competitions in which they have looked at prediction of structure, but also prediction of interactions. Now we'll come back to that later on. And in these competitions, actually they validate different tools, different servers. And actually, in this paper, they describe how good is automated protein docking, and you see that the quality is actually becoming very good of this automated docking servers. And we actually tried that also during the day course, and we tried to have a look at a number of existing interactions that we know of. We upload the separate molecules on this docking server, and then we actually validate how good it predicts. In most cases, actually, you always find, and let's say the top 10, in most cases you are able to identify the true site where the docking is happening, where the interaction is happening. So this is actually the one that we are using in most cases, which is actually a very good state of the art in this competition, but there are also other ones. So if you would want to try this yourself, in one of the workshops, actually this is discussed, the Plus Pro docking server. So I will come back to that in one of the workshops. So what you can do, of course, is for this, you only need 2 PDB files to upload. One of your ligand, smaller molecule, one of your receptor of the largest molecule. You upload these 2 PDB files, and then the docking server predicts a number of conformations how the interaction can occur. So these PDB files, these are coordinate files. Can be experimentally determined structures, but some cases, if you work on a specific protein, maybe there's no structure available. And what you could do is, if there is a good homologous protein structure available, you could use that one to predict or make a model of the protein you are working on, and you could also use that for docking. So that's 2 parts. You have to then predict the structure of the protein you are interested in, and then when you have a coordinate file, you validate, of course, the quality. For example, a Ramakandan plot, so a number of ways how you can check the quality of your structure. So you can also do that for a homology model that I will discuss now in this chapter. Then, if you would want to look at an interaction, you would also use such PDB file, once you have the coordinates, to have a look at certain interactions. So that's also possible. So this chapter now, this is the largest chapter for today, is about structure prediction. Why it is important? There are about 150-160,000 structures known. But it can be that you are working on a protein that there is no structure available. So then of course you have a problem. What you do, you can't visualize the protein in final. Chances are that actually the domains that are present in your protein that you're working on, there is a similar structure already available in PDB. And you can actually use that one to make a homology model. And this is what I will try to explain in this chapter. There are three ways how you can do or predict the structure that is shown here. So the first one is you try to guess if it is a new fold or it is an existing fold. Because if you know it is an existing fold, then you can use that information and understand the protein you're working on if you know the fold. So this is called fold recognition. The second one, if there is already a structure available in PDB, then you can actually have a look at homologous structure and do comparative modeling. So comparative modeling, or what we call homology modeling. Let's say you want to have a look if this is possible, homologous structure in PDB. What would you do? You have your protein sequence, what would you do to find out? Line it. So which tool would you use then? BLAST, indeed, yeah. So on the normal BLAST website of NCBI, if you look at which databases are available, the PDB will also be available there. Or you can go directly to the PDB website and then advanced search. I think I showed you that last time. Advanced search, and then one of the options is also FASTA BLAST to have a look at if there's a protein available in the database that is homologous to the one you are interested in. So that's homology modeling. And then the last one is if there would be no information available, if you cannot know if the fold already exists or there is no homologous structure available in PDB, then you can do what they call ab initio or de novo prediction. This is of course the most difficult one. But I will show you that the results can also be quite nice. Now if you want to do structure prediction, there are of course again tools you can use. Also for interactions there are servers that you can use. What I'm telling you today might be that in 2-3 years, state of the art is different. So you have to know if you would want to do such thing in 2 years. If I tell you now use the Plus Pro server, maybe that's not the best one in 2 years, right? So how can you know what is at the time you want to do that state of the art? Then you have a look at the competitions. So there are competitions worldwide where people that are experts in this, in structure prediction and interactions, they compete in this competition. And then these results are published. If you look at the results, you know what the best one is at that time. That is the way how you do it. And then you can choose a tool, for example. So which competitions do you have? You have the CASP. Looks like CASP from caspase proteins, but actually it stands for Critical Assessment of Structure Prediction. This is where still some manual work is being done, which means people are actually looking at this and trying to predict the structure. But you can also have it fully automated, which means you just upload a sequence to a server and the server will just predict the structure. This is the CAFAS, or Critical Assessment of Fully Automated Structure Prediction. So that's the difference. How does this work? Actually, a couple of months before they start, they already know that structures will be available. So let's say if I would have a protein and already have a crystal, I know it will diffract. I'm actually solving the structure. That's the perfect target for these guys who are doing these competitions. Because in a couple of months I will actually have the experimentally determined structure. And then I can compare it with what they predict. So this is actually how this is done. Yes. So the difference between CAFAS is that these are all predicted and done automatically. And then of course you get a ranking, and then there's a conference where they discuss the results, and then you know which one is the best and so on. And the last competition was actually in December 2018, so I think this one is every 2 years. And the website there is predictioncenter.org, so if you want to have a look at those results, you go to this website, you click. So in 2 years there will be a new competition, in 2020 actually. Probably at the end of 2020 there will be a new competition, and maybe there will be already some better tools that you can use to predict structures. A similar critical assessment is CAPRI, which stands for Critical Assessment of Prediction of Interactions. So that's not only predicting one structure of one protein, for example, but you're also looking at how different molecules might interact. And this is actually where I refer to the paper of the ClusPro server, how good is automated docking. This is where the ClusPro came out very well actually, and why, if I would have to do automated docking, why I use the ClusPro server based on the results of this competition. There is also a website at the European Bioinformatics Institute, EBI, CAPRI website, where you can find the results published there of this critical assessment. Okay, so homology modeling, I think, is something you can actually do or should be able to do. So I will spend most time on this part, and then I will also discuss the fault recognition and the ab initio, the two other methods. So why homology modeling? Of course, if you want to know how a protein functions, you want to have to know and understand the structure. How can you do homology modeling? On what is it based? Actually, it should be straightforward. If you have the amino acid sequence and you know all the theory, how structures work, it should actually be in theory sufficient to acquire the structure. The second point is, of course, during evolution, that a protein is more stable than a sequence. What I mean is, you can just keep on changing the amino acids in a protein, so the sequence similarity will drop, but it is still the same fold and the same function. And this can actually be quite extreme, so you can have less than 20% sequence identity, and even by blood you cannot identify matches between two proteins that they are exactly falling into the same protein and functioning the same way. So that's possible. So this is what this part is talking about. Similar sequences can adopt a similar structure, but also distantly related sequences can have a similar fold and a similar function. Of course, you have to know and try to find these homologous sequences to be able to do homology modeling. So this is, let's say, a plot that you can use to have a look what is possible. So you see here the number of aligned residues. So it only goes to 250, but you can just actually continue here, more or less. And this is the percentage of identical residues. So sequence identity, not similarity. You see here, if I would have a protein of 150 amino acids, and the sequence identity is 50%, which means I'm here, where the X is, I'm in a safe homology zone. Which means if I find in the PDB database, for my protein of 150 amino acids, I find a structure which shares 50% sequence identity, you should be able to build a reliable homology model. That shouldn't be a problem. And you can already see in most cases, let's say the threshold, let's say, is between 20-30%. You could say 25%. In most cases, I say above 30, it's safe. So above 30%, it is possible, especially if you use more than one template, more than one structure, it is possible to build a model and get an idea of how the protein is built. If the protein is very small, then it might require a higher sequence identity. For example, 100% you see, 30% is, let's say, a bit on the border here. If it would be smaller even, you would require a higher sequence identity. But in most cases, of course, proteins are much larger. And I think if it's above 30%, it shouldn't be a problem. And it is possible to have at least an idea of how the structure looks like. And then this is illustrated actually on these slides, on the next slides, what I just explained to you. Then how does it work, the homology modeling? Which steps? There are 7 steps in total. And this is just the first one that I discussed now. You have to find which structures you can use. So this is what we call template recognition, and a simple blast is the first thing you would do. Go to the PDB website or on NCBI blast, you choose the PDB database, and you just look if there is structural information available. If you get one hit, that's already good. If you get multiple different ones, that's even better. The more you have, the more structural information you have. And this will actually improve your alignments. So if you would look for a certain structure already with the blast, you will get an initial alignment. But this is just a sequence alignment. You always have to correct your alignments. I will explain later with a nice example why this is. Because it doesn't mean if you would have 2 exact residues, pro and proline, you would say an alignment, it's a match, sequence is just the same, the residue is the same. That doesn't mean in 2-dimensional space that they are exact the same position. This is why you have to correct your alignment. So it's also possible to do 3D alignments based on the structural information on the coordinates. There are tools that you can use to do that, and I will show you 1 or 2 such tools that you can use to actually get a correct alignment. So this is actually very important. This is quite easy, just a blast, but this is quite important. If you get this right and good, then it's actually very easy to do homology modeling. There are very good tools. I always use Modeler. If you get your alignment correct, then it's quite easy with Modeler to actually generate a homology model. So this is what you do, and then this is actually what the tool will do for you. So you have little work on these steps. Backbone generation is what Modeler, for example, as a tool will do for you. How does this work? Well, it's simple. From your node structure, you know where your alpha carbons are, right? So this is what we call the trace. If your sequence will be exactly the same and it follows the same trace, it also contains all those residues alpha carbons. So you just could copy-paste the position of these alpha carbons. This is actually generating your backbone, the path of your backbone of your sequence. So this is the first step. Once you have that, it's just knowing where the loops are, knowing where the side chains are. So this is why these are the next 2 steps. You try to model the loops because often these are more flexible. So this is a bit more difficult. And also modeling of the side chains. But these are, again, you have some tricks to help you with this. And then the last part is, of course, validating this, typically in iterative, trying to optimize your model. So this is 3 to 6 what Modeler as a tool, for example, can do for you. 1 and 2 is what you do. And of course, this is also very important. This is what you have to do again, is the model validation. As a result here, if you would, let's say, generate 10 models, you can already get a certain scores, which will tell you which models are reliable and not. But, again, in the chapter of structure, quality and validation, there are some tools that you can use to have a look at this. And again, what would be one such an example? Which tool that you would use? You would look at the VMC angles, so which plot would you make? A 100 plot, indeed, that would be one thing to do. And you saw another tools, a few other tools that you could use to try this. So for example, B factors and other things like that. You could also have a look at that. On the next slides, actually all these 7 steps are just explained in more detail. And some of these things I think you already know. This is then with a figure explaining this. So here, selecting your template. So then this is the target, only partial And then here you can do tap identification alignment and then start the backbone generation, loop and sidechain modeling, and then you optimize your model. Now, how does this work all in detail? Of course, to align, you already know the BLAST algorithm. And I think in another course, you either already saw it or you will see it, how these residue exchange matrices work. Palm, Blossom, does that ring a bell? Yes? Okay. So then I can briefly go over that. So this is here such residue exchange matrix, then where you get a certain score. Of course, if it's a similar residue, it will get a very good score. For example, here, phenylalanine, if you would have tryptophan of tyrosine, that's a good score. If it would be, for example, an arginine with a phenylalanine, that will get a negative score because these residues differ greatly. And then based on that, you can make your alignment. So this is then how an alignment matrix works. So the first sequence, the second sequence, you have all these scores depending on the matrix you choose. And then you actually choose the path with, of course, the highest score. So in this case, for example, here you see a shift. And then this is, for example, where you would have a deletion or a gap, actually. So this is representing a gap. Now, this looks, I think, logic, but maybe this is not. If this would be your two sequences, it's what we call the pathological alignment problem. You have a sequence LT, 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 LT. You have a sequence YA, YA, YA. How would you align these? Impossible to know, right? With only two sequences. Actually, this is the correct one, the top one. How can you know this? If you would have a third sequence. And this is why multiple sequence alignments are very powerful and useful here. If you would have a third sequence, TY, 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 you can actually align the tree, and then it makes sense that the T corresponds with the A, and the L corresponds with the Y, so that this would actually be the correct one. So this is why multiple sequence alignments are, of course, very useful. And this is actually quite good for your initial alignment. To improve your alignment, I would advise you to use also 3D structural alignment tools. And these are two tools you can use, Matlas and FatCat. So I added the websites, because if you Google for these, you will probably end up on, you will never find the website. And actually they will, so you also upload PDB files of your templates and they make structural alignment for you. And that will help you improve your alignment. Yes. And this is actually a very nice slide to illustrate this. So the dark one here is the template. Right? So this is the one you find in the PDB database, so you know that structure. And this is also showing you the dark one. So you have 13 residues starting here, 1, 2, 3, and so on. And then what a normal alignment program would do is actually with your model sequence, it would generate a bad initial alignment. Why? Shown here, cysteine matches with cysteine, arginine with arginine, and then this is still similar, hydrophobic, and proline, this makes sense. And then it will introduce a gap of 3, and then Clu, Ala, and so on. This makes sense if you just look and think in sequences. This is how a crustal, for example, will work. But have a look in 3D. Is this really possible? So you see here at position 5, we have here the arginine. So that will locate it here. And the next one where we have here is position 11 is here. So if it would be indeed this, the correct alignment, if you would have these matching and the proline of your template would be here, position 7. And then the gap of 3 would have to make the distance for the next residue to position 11. This distance is far too large. It's never possible for these to be two neighboring residues. This is impossible. So what a structural alignment will tell you is that this one is actually good. Why? Then you introduce your gap here. 1, 2, 3. You skip these 3, and then actually you again start at 9 at the alanine. No, sorry. Here at 11. So then you will have your distance here. So these two will match here, although it may not make sense. Serine, alanine, alanine, proline, you would say, but this is feasible in 3-dimensional space. And it would just mean that you could just keep on copying the alpha carbon atoms of these, and then of course your side chains might be different. But this is possible. Why? Here you have residues, but in this case you have a lot of space here. So you could fit your residue side chain of position 9, in this case an alanine, you could perfectly fit it here because you don't have this residue 8 anymore. How does this work in a protein? In most cases you would have a secondary structure here, a loop over, a strand or a helix here and here, and in most cases what you see is that loops are either longer or shorter. This is actually where the differences are. And also we will make an alignment and you see gaps or insertions. It's always at most cases at the top of your protein, at the surface, and it's just certain loops that are longer or shorter. This is actually such an example. And this I think illustrates quite nice why a structural alignment can actually be better and help you predict the structure of the model correctly. And of course, if you do that, you can construct your backbone. So in this case, what I just do is, from my template, I copy the backbone, which is actually just shown here. So in this case, this would also be the backbone of my model here. And then the next steps are the loop conformations. How can I do that? Loop conformations, there are two approaches, knowledge-based or energy-based. How does this work? Simple, knowledge-based. Of all the high-resolution structures that already are in the PDB database, you extract all the loops and you just look if it's a similar loop that already exists. And then you can copy that information from an existing loop. So actually, loop conformation copying is supported by all the tools and servers that do homology modeling. So that's knowledge-based approach. The second one is energy-based approach. Less is more. If it requires less energy, your protein is more stable. So this is from one paper that I used this figure to illustrate that, where you see here a number of loop regions, and then here you actually see the score. So you see the lowest score is loop 7, which is then probably the most feasible one. So this is then using an energy-based approach. For the side chain, similar. How can you do that? You have certain rotamers, and you can use, again, existing knowledge, a knowledge base of existing rotamers. What are these rotamers? This is a nice example that illustrates this. So for this side chain, you can actually have this position or this position, which means you have two different rotamers. They can be rotated here the way they stick out. And in most cases, what you see, even if you would have, let's say, lots of possible conformations, in most cases you see that there are only a few that are always being present. So in this case 2, but often it might only be 1. And this, of course, if you use such library of these rotamers, known rotamers, it will reduce the number of calculations you would have to do. Why? If you would have 100 amino acids and you would have 5 possible rotamers, so let's say 1, 2, 3, one in the back, maybe one other one, 5, it would mean that your computer would have to combine all of this and calculate 5 to the power of 100 combinations. That's very high demanding, of course. While if you would only have to maybe 1, 2, compare that, it's computationally less demanding. So that is why a similar approach, you can again use known knowledge of all the structures that we already have, compose such rotamer library and use that information. So in this case here, 2 possible ones, and then you see how would you choose one of these. in this case, how would you choose one of these? What could be one thing? And then please think not only at this, but at the whole space in 3D.
What's most likely to react with other proteins?
Yes. So
The one with most.
actually, there can also be things here, of course. It could be another protein, but it could also be, of course, that your protein here continues, and it might be that there's another loop here, that will be one here. If that is indeed the case, and you would have two rotamers here again, it makes sense that this one, they would collide. It would be structural hindrance. So then it's obviously that it would choose this one. But of course it will depend. That is why, of course, you have to look at the whole structure again, and that you will optimize again in an iterative way and look at all the positions to combine this information, of course. Yes. And that is this step. So model optimization requires, of course, an iterative approach. You predict the rotamers, have a look if there is a shift in the backbone maybe, and then again, so on until convergence is achieved. And you can combine it, of course, with energy minimization steps until you get, let's say, a stable protein in total. You already know there can be also errors in experimentally determined structures. So, of course, it can also be possible in your model. That's why the last step is very important, that you look if there are errors in your structure. So the Rampage tool is one that I like to use. It generates very nice Ramachandran plots. Another one, what you can do is the project report, generate that with a tool like PDBsum, and that will also tell you not only Rampage Ramachandran plot is only the two of the phi and the psi angles, but you can also have these angles in your side chains, and this tool can also have a look at that. Now, of course, once someone already uploaded a sequence and asked for, on a server, to ask for homology modeling, if you would upload the same sequence, the server does not need to calculate that twice. That's why the two most used tools is actually SwissModel and Modeler. They also store this information in a database, in a repository. So you have the SwissModel tool, which you can use for fully automated protein homology modeling, which means you just give in your sequence and it will calculate a model for you. This one, Modeler, you can actually more optimize it yourself. Provide multiple templates, optimize the alignment. So you have, let's say, more features or more parameters you can set for yourself with this one. And this is actually during one of the workshops with today's students that we try to do that, and this is in one of the workshops that I will discuss later also this evening, how you can make a homology model of yourself with Modeler. So this is the website where you can download the tool. This is how SwissModel looks like. Then you can here do modeling or have a look at the repository of the models that already have been made. And this is how the Modeler website looks like, where you can download and you then just ask for a key. So you need a license key, but it's always the same license key. And then you can install this program on your own laptop and actually also make these homology models on your laptop. So you don't need a very powerful server to do that. It also works on your laptop. These are actually the tools that are all available. So you have the Modeler tool, but also ModLoop are then, for example, for specifically only having a look at the loops to model. And the ModBase here is actually the database, so the repository. So that's important for you to know the difference. So on the SwissModel website, here you go to make your own model. And the repository is the database of stored models. And actually, if you would click on this, you will see that there are already quite some available. for a number of species. Maybe I can quickly demonstrate that. Swiss model, the repository. So then here, for example, you see Homo sapiens, proteome size 20,950, sequences model 17,000, and then models 43,000. Why is this higher? It can be that you have multiple models for the same sequence. So it could be that you ask to model a sequence in, let's say, 2005. It uses the structural information and templates before 2005. If there would be a better structure of a homolog, let's say in 2010, and you would ask today to make a new model, it will probably be a better model because there is new structural, experimentally determined structure available. So it could be that a new model is being generated. That's one option, where there could be more. Or you can have multiple models if you would have multiple domains in your protein. So you could only, let's say, if you have two domains, only make a model of the internal part of the first domain, and then maybe also a separate model of the last part of your protein. That's also possible. Anyhow, it's already quite impressive that for lots of the sequences or the proteins and the proteomes that models have been made if you look at all these model organisms. So that's already quite impressive. And the same then is the case for modeler, and then you just go to modbase. I think you can also find the stats there. Modbase, I will add Salina. Yes. And I don't know, do they have the statistics here? Here's statistics. Yeah. So then this would be the same. Homo sapiens, here you see actually much more. Here you see that they work on a transcript base. So if you would have, let's say, alternative splicing, they work here, number of transcripts, 170,000. Then you have models, 140,000 transcripts of these. And then again, here you have, of course, more models. And then these figures are from 2016 now, so there might already be more available. Yes. So that's it for Swiss model and modeler and their databases, their repositories where the homology models are being stored. So that's it for for homology modeling. So I've told you the 7 steps, how these work, which tools you can use, and where models that are already been built, where you can find those. So then the 2 other things I want to tell you about structure prediction is fold recognition and ab initio, the 2 other methods actually. So there are different ways they call it. They have also been called inverse folding problem, threading, fold recognition problem, but it's all the same. And again, they make a distinction in methods sequence-based, methods energy-based. So no information and energy-based approaches. So no information. Why? There is, of course, lots of domains, families that are being overrepresented. I thought there was a slide here telling where, yeah. So if you would look at all the protein superfamilies that we know, actually globally about only 100 folds represent half of all the protein superfamilies. So that's not a lot, about 100 folds. And you see that lots of same folds are being reused by nature. And this is one such fold. The Greek key topology, if you remember it from the protein chapter in the beginning, you have 3 strands next to each other, and then the third one is next to the first strand. Sorry, the last, the fourth strand is next to the first one, so which is typical of Greek key topology. So you see that here in an antibody, so you have it in immunoglobulins, but for example, adhesion molecules also have this fold. Why is this? So we have several possible explanations for this phenomenon. 4 I will discuss: divergent evolution, convergent evolution, just because there apparently seems to be only a limited number of possible folds, and the last one is actually wrong analysis. Let me first talk about divergent evolution. So proteins with similar folds are of course related, but it can be that sequence analysis tools are not sensitive enough. So if they diverge enough during evolution, you can actually not find it again if you just would blast them. The sequence identity is too low, which I already told this evening that it is impossible to find at the sequence level, but that the fold is still the same and the function is still similar. So a normal blast might not detect it. That's why there are already better, more sensitive algorithms. So one that you could use is the position-specific iterated blast. I think that was also discussed normally in another course. And then the hidden Markov models is also a more recent algorithm, better than a normal blast, which might be used to actually to find less or sequences with less sequence identity, still be able to detect them with the psi-blast and the H&M search. So this is divergent evolution. You can also have convergent evolution, that just certain proteins that nature actually comes in a separate way to the same structural solution. So you actually only need a small sub-fragments to be able to have the active site. And one such example, very well known, are serine proteases, where you actually just need the active site to be available with only a few residues that need to be conserved, but it can that the rest of the molecule might be a bit different. So it's just you start actually from 2 different proteins, but you come to the same structural solution to have, for example, a protease function. This is convergent evolution. And then the limited number of folds is that many of the theoretically predicted combinations of secondary structural elements, you can actually not see them in nature. You cannot find them in nature. So it seems to be that there are only a few possible ways to actually compose or be able to build a fold. So that's also why the ones, the supersecondary structures that we talked about in the first chapter, that they are also being reused. So you have the Rossmann folds, 4 helical bundle, and a number of other ones. This is also again a combination of secondary structure elements that are just being reused in different proteins. So that's a limited number of folds. And then wrong analysis. I talked about the SCOP and the CAT data bank. I don't know if you remember what these databases are for classification of structures. If you compare them, they actually only agree in about 66% of the protein chains, which means you have a structure, you look it up in the SCOP database, it is in one kind of classification. It could well be that in the CAT database it's in another part. So another type of classification. So this means wrong analysis. Why? Why is there a difference? So it seems to be that you can have, let's say, false positive similarity, so that's possible between 2 structures. They differ from other structures and they're actually not really comparable. So in this case, prediction of similarity is not feasible and very useful. So that you can find in these SCOP and CAT databases. So of course, still 66% is more than half. So most proteins normally do have the same classification, but in some cases it's known that the analysis is wrong and that the classification is not entirely correct. Fold recognition can be successful in the first 3 cases, of course, which makes sense, not if the analysis is wrong. But in these 3 cases, you should be able to find the fault, recognize the fault. Then you know the fault, and then you only have one question. Of course, what is the function? Is it the same function, or does the protein, even if it has the same fault, does it have a new function? So if the fault comes from a homolog, chances are that certain parts of the function are preserved. One thing that again you could do is, you could have a look at a multiple sequence alignment. Where will the residues be conserved most likely? And your active site. If the residues that are important for the function of the active site are conserved in the fold you recognize, then probably the function is of course also the same. If residues are mutated in the active site, then the function might be different. So that's, for example, one thing you can look at. You could also look, certain molecules need cofactors. For example, the cadherins, during the exercise you saw they have calcium, but you also have proteins that need zinc. You could have a look at those residues are conserved. So these are the things actually that you can look at if residues are conserved or not, and then draw certain conclusions if the function is same or not. Yes. If you want to try this for yourself, what are the servers that you can use? Here are the 3 servers that you can try, and they actually will try to predict the fold of your protein. So that's fold recognition. And then the last one is ab initio. So if the other 2 fail, you can't use another experimentally determined structure as a template to make a homology model. You can't predict the folds with fold recognition, then you have to use ab initio or de novo structure prediction. And this is of course the most difficult one. One well-known method is the Rosetta. I don't know if you've heard of Rosetta at home. Do you know Rosetta at home? No? No one? Do you use a screensaver? Yes? Actually, if you use a screensaver, your computer is doing nothing, right? So this initiative actually, if you would, I think you have to install something. If everyone in the world would install this and would provide his computer when it is in screen saving mode, it could be used to make calculations for these kind of predictions. And then you would have one huge supercomputer. That's actually the idea here from Rosetta at home. That you share, if you do not use your computer, your resources of your computer to calculate these kind of structures. That's the idea of Rosetta at home. And these are actually some results of Rosetta. So they also competed in the CAST4 critical assessment. And then here you each time see what the prediction was of Rosetta. And then this is actually the native structure, what it was. You see often that the fold here, maybe it's one long loop, 2, but even if no information is available, it's still already quite impressive that it is at least similar. Maybe not the side chains are not exactly the same, but I think it's obvious that you can recognize the fold, that you can recognize the secondary structures. So I think that's already quite impressive. Now, why are these methods interesting? What are the possible applications? Actually, for functional annotation of genomes and structural genomics. So let's say if you would have a new species you sequence, you do a gene prediction, you know all the genes, you know then the proteome or the theoretical proteome, and then you can actually have a look at all possible proteins encoded by this species. How you do that? You can first have a look of all these proteins that can be encoded by this species. Can I make homology models? That's the first step. And there are quite a lot. You will be able to already build models. Then you can go also to full recognition and then also to ab initio. And in this way, from any new species you sequence, you can already have an idea how the protein and the structures of the proteins look like by using these applications. Even if you did not determine a protein experimentally, the structure of this species. So with these methods actually you can do that. So like you can do genome annotation and predict where the genes are, you can also predict all of the proteins, how they look like of a new species you sequence. So and then this is explained here. And again here some results of that. And then there's one extra part of structure prediction. This is actually just a simplification, but there are some existing tools that are very useful and very reliable, and it's what we call prediction in one dimension. How does this work? Because we have three-dimensional structure, x, y, z coordinates to know where they are. How can then we predict the structure in one dimension? And the best methods can actually predict the basic properties of a fold in some cases. Why is 1D becoming more and more accurate and important? It's actually a simplification where each residue, for example, in 3D structure, you actually assign a string of structural assignments in one dimension. How does this work? For example, for an amino acid, you can say, is it part of a helix? Is it part of a strand or is it part of a loop? So if you would write down the amino acid sequence, below the sequence you could already say, is it an H, a helix, is it an S, a strand, or an L, for example, for a loop. And in this way, below your 1D, remember the primary sequence, below that you could just write in also 1D then, just H, 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 L, L, S, 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 like this, a string, assigned a string, and with that already know the secondary structure, right? So this is how we assign a secondary structure represented by only one symbol for each amino acid. So that's one way we can do it, but another approach could be using a number to indicate the accessibility. So again, you could use a score from 0 to 9, depending if it's more on the surface or more on the inside of the protein. And then again, below each amino acid sequence you could write 0, 0, 2, 3, 4, 9, 9, 9, and so on, telling you where certain residues are more likely to be on the surface of your protein or more on the inside of your protein. So that's another way how you can project that in one dimension. So such strings of per residue assignments are what we call 1D. What are the three very popular methods that we can find? Secondary structure prediction is of course this one. Solvents accessibility is another one, that's this one, and there's a third one. Third one is transmembrane helix prediction. Why is this important? This can be important if you want to know, is my protein in the membrane of my cell, for example? Might it be a receptor? That's, for example, very important if you want to do certain experiments. Another way why this can be important is, let's say you want to develop a vaccine for a new virus. You want to know which proteins of the virus are also on the surface. For example, for influenza, which targets the vaccines. Most of the vaccines you get each year, I know if you get a flu vaccine, they target two proteins. Remember they assign them H1N1, H1N5. No, doesn't ring a bell. So the H is for hemagglutinin and the N is for neuraminidase. And these are actually two large proteins on the surface of the influenza virus. There's also another smaller protein, the matrix protein. And actually they are now working in Ghent on a universal flu vaccine targeting this small protein, because that does not mutate that often. Actually, almost not compared with the two large proteins. Each year they just keep on mutating, and that's why each year, also because it might be a different strain, each year you actually need to update your vaccine, let's say. So you want to know which, of course for influenza we know it, but maybe for another one, you want to know which proteins are on the membrane of the virus. That could be another application. Transmembrane helix prediction. And this also works very, very good. Yes. And here are some tools that you can use to do that. So you have Predict Protein, Cypret, SS Pro. This one I think you can also download and run it locally, which means this is good if you want to use one sequence, but of course if you would be able to download the tool, you would have 10,000 proteins. You could just, with a script, do a prediction on also tens of thousands of proteins. And in most cases, just pasting the sequence and that will tell you the results. And I know if I have screenshots of these results. Yeah. So there are each time different variants. For example, this one, the SS Pro, you have actually two versions depending on how many classes you have. So the basic one, secondary structure, for example, 3 classes is just strand, helix, loops, but you can have more classes, 8 classes. Also for the solvents accessibility, 2 different options, and of course the more you select here tools to predict, the more or the longer it will take to predict that. So besides the secondary structure prediction, here you can also already do the solvents accessibility, but also other tools like disorder, which I will come back to in the last chapter. And then this is just a number of tools showing you that you can use pasting the sequence, and then this will do the assignment in 1D. One thing I want to mention is here. So once you know which part, if it is a membrane protein, and which part is in the membrane, that's illustrated here. This is the inside of your cell, cytoplasmic, and then this is extracellular in this case. This is representing your transmembrane helix. What will this be? This will just be like 20 hydrophobic amino acids. Maybe 2 or 3 other ones, but in most cases, many, in the range of 20 amino acids, many hydrophobic residues. So then it could be that you have a protein with only 1 transmembrane helix, could be 2, but it could also be more. You have proteins that have up to 7, for example, I think GPCRs have 7 transmembrane domains. What is then important to know is which parts of these proteins are on the inside and which parts are on the outside, right? So in this case it's difficult. Inside here, or it's just the opposite, which is quite important if you want to develop a vaccine. You of course target the ones on the outside, not on the inside. So that's important to know. And how can you do this? Just by looking at the sequence, I can tell you. Quite amazing. There's a simple rule. It's called the positive inside rule. And it was already developed quite some time ago. Here, Gunnar von Heijnen in the late 80s. If you have a look at, first of all, you have uppolar between 20 and 35 residues, but in most cases 20. Then you just look at some amino acids before the helix, the sequence, and afterwards. And you will often see that in one part, either before or after, you will have more R and K, more arginines, lysines. That one is on the inside. Positive residues on the inside. That's the trick. So by just looking at the sequence, you should be able to do that. And that is actually illustrated here then. So for example here, in this case, you would have a few more R and K amino acids in this region. and then you would have it here, but not there and not there, for example. So that's a very easy trick to have a look at that just even by looking, but of course these tools also take that in consideration. So there are also a few very good tools to do that. I like to use these three. This is already quite an old one, TM-HMM2. Then this is actually, I think, of the same people of TM-HMM2 Phobius squad, and then MEMSAT 3. One thing that is often difficult is if the helix is at the beginning of the sequence. Why is this? Because it can be, if it is in the, let's say, the first 30-40 amino acids, it might be that it's a signal sequence. So that actually your protein is just being secreted. So it needs to go through the membrane outside, extracellular. And then often at the beginning, you have also again hydrophobic residues, which is actually a signal that your machinery in your cell knows I have to secrete and transfer this protein outside the cell. And some of these tools, and I think the Phobius already takes that into consideration and will tell you this is actually a signal peptide then. And then I think there's a list of a few other useful tools for specific things. For example, also transmembrane beta barrels can be detected, but these are specific, let's say, applications then. So the three I discussed here are what is being used the most. And then this was an initiative actually developed a database already in 2011 by VIB, the BRICS database, but it seems to be no longer online available. But the idea was very interesting, that you again, like you have these libraries that you can use for modeling your loops and things like that you do that for all high resolution structures. You just split it into smaller fragments. Then you have a library of all fragments peptides actually. And you could use these to build like you use with the Lego blocks. You like build something, a house or whatever. You could use the same bricks of protein fragments to also, for example, use it for structural work. That's the idea after this database. So that's why it's still in the course. I think the idea is very nice. And then here, for example, you would have, let's say, you see here a class of a number of fragments, and then this is where you get the sequence logo. So in this case, most of the part is helical. So this would be this one here, helical, with then here part of a loop. And then you can have certain classes, fragments, and so on, and use these building blocks of peptides to build, let's say, a protein. Okay, that's it for this chapter. That's let's say, the largest chapter, and then the last two are very small, so also not that much theory for that one. So for this chapter that I just discussed, the structure prediction, the most important part is understanding the theory, the three methods. You're able to explain the methods. For example, also understand the steps in the homology modeling. You will not have to do docking or homology modeling on the exam, but you might be able or need to use a tool. So that's actually part where you have databases and tools. Could be that it asks you to do a certain, just use a certain tool. So you need to know which tool to use in which situation. Let's say if I would ask a certain question, then you know, that was there in the course, so that's of course the open book part that you know. For example, if I would ask you, let's say, can you tell me if this protein would contain a transmembrane region, then you immediately know, I can use Phobius or TM-HMM2, for example. Or if I would ask, can you tell me the secondary structure prediction in 1D, that you know what that means, that there are tools, just if I would give you a sequence, that you can and tell me that. Okay, and then the last two, structural genomics. So structural genomics were two initiatives that already stopped a few years ago. Now, of course, I already told you we have homology modeling, fold recognition, ab initio prediction. So if we would have a new genome, we could use those tools. But at the time, there were actually two initiatives that tried to determine in high throughput, large-scale structure prediction. That was the purpose of these structural genomics projects. So there were two projects from 2002 to 2007 in Japan, which was called the Protein 3000 project, and there was also one in America, in the United States, from 2000 to 2015, which called PSI, or Protein Structure Initiative. So you have to know these two projects then, and just understand, what they actually do, why they are useful, and also maybe advantages, disadvantages, but not the details of this project. So, for example, not all of these details. Again, I refer you to the databases where models might be stored, and then here you just see actually some shots and some results. So you can just read these. So this is maybe interesting just to tell you. This is actually how they work. So first of all, you select your target. So can I make a homology model or predict a structure? Yes, then of course I don't have to choose the target. If it might be a new one, and if it's an interesting one, then I choose to clone it, to express it, purify the protein, and crystallize it, of course. But this is not that straightforward. You can have all kinds of problems. One problem could be in E. coli, for example, it does not express, so you might need another expression system. If that does not work, of course, you just drop it. In case it does work in another expression system, yeast or maybe insect cells, you continue, try to purify it. If it can't be soluble, you might try to solubilize it, refold it, and so on. You try to purify it, and then of course you want to crystallize it. These structural genomic programs at the time were of course also really useful for these critical assessments, because they already had, let's say, somewhere here, they already knew which structures will become available. So they actually provided lots at the time targets for these critical assessment for these competitions in structure prediction. So that's also something to remember. And then, of course, once you have purified protein, then of course you try to crystallize it, and you try to solve the phasing problem. You need to add selenium, and you have selenium net crystals and so on, and then try to build your model, refine it, and then of course deposit your PDB structure. So this is the flow chart of these structural genomics initiatives. And then here you have just a case study. You can also just read this. This is also still interesting for you to know, because this technique is discussed here, but you can use it in general when you determine a structure experimentally with X-ray crystallography. It's called dynamic light scattering. And this is important once you have your protein to know, is my protein now good enough? Can I use it to make crystal? Because you might try making a crystal, spend months of time, lots of money, that it will not crystallize. And that could be that there's an obvious reason. And you might find that reason by just doing dynamic light scattering. So how it works is it measures laser light scattering in your molecules in solution. So that's actually how it works. So it actually determines the size and also the distribution of the molecules in your solution. So the polydispersity. This is such an example of such a machine, so dynamic light scattering machine. This is showed the principle. So in this case we use laser light, a lens shoots through the sample, detects the data, and then you get such scattering pattern. And it can look something like this, or it can look something like this. If you see this, then this makes you very happy, because you have a monodispersed solution, which means your sample is homogeneous, and this should be easy to, not easy to crystallize, make it doable to crystallize. While if you would see this, this will be very difficult or even impossible to crystallize. Why? Because you have different kind of molecules. So you have a polydispersed solution which is non-homogeneous. So different sizes could be that this is, for example, the monomeric form of your protein, and this could be ditrimeric form of your protein. This will not be able to form a normal crystal. So for example, then you could say I will do a size exclusion chromatography to split these in which the size that matters, and that maybe in one fraction then you will get this P. You do DLS again, looks like this, okay, and then I can use this solution, protein solution, to start my hanging drop, vapor drop diffusion, if you remember. Sitting drop and hanging drop. And then try to crystallize it. So DLS, dynamic light scattering. And the last thing I want to mention in this chapter is this here, because the results you can say are quite impressive, but there's also a drawback, of course. So at the time, lots of new structures became available from these two initiatives. So you see here in 2006, 2/3 of the global structural genomics data came from PSI, so the American initiative. 20% new protein families came from that initiative also. Also a lot of new classification, folds and superfamilies came. But what was the drawback or the negative part of this? First, it costs a lot of money. $600 million. And what do you get? XYZ coordinates, basically. Just coordinates, PDB files. This is what you get. Well, simplified, of course. So what is the criticism? You don't know anything about the function of the protein. If you would spend all of this money divided into research groups, they will try to determine the structure, but also have a look at how the protein functions, maybe try to do an assay or another kind of experiment, get more information that not only, let's say, the structural coordinates. So this is, let's say, one of the criticisms on these two projects. But still, we got lots of new, nice data. If you want to make a homology modeling, then of course, you could use those templates to build them. Okay, so not that much in this chapter. Know the two initiatives, know to explain them, know the DLS, and then positive, negative things about these two initiatives. And then the last chapter. So you know how you can determine a structure experimentally, where you can find this information, PDB database. If there's no structure available, how to make a model. So we're all talking about structure now, but actually there are also proteins that are not structured. This is actually, I think, why this is also important. This last chapter is talking about intrinsically unstructured proteins. And I'm sure there's at least one protein that you will know at the end of my talk now that is actually quite unstructured. So for a long time we actually thought they had to fold correctly to be functional. Now we know that this is not always the case. For example, you probably all know P53 tumor suppressor protein, TP53 or P53. A large part of that protein is actually unstructured. That we know now. So more recently we found that not all proteins function in a folded condition. So there are different states, let's say. Some proteins are unfolded, or we can also say disordered or unstructured, and they can still be functional. So what does this disorder refer to? It's actually looking at the equilibrium position of the backbone. Is it stable or not? So we can look again at our, remember the dihedral angles, if they have a certain specific value, or it may be that they vary in time, so that they're not always at the same position. So that's why then they are unstructured, they are flexible, not always at the same position actually. So proteins, these unstructured proteins, are actually proteins where it can be the entire protein, but in most cases it's just part of it. So contains regions where stable tertiary structure is lacking. And you have protein of 20% to 80%, so it depends on the protein. There are three different terms you find. Some people call them intrinsically disordered proteins or IDPs. Some call them intrinsically unstructured proteins or IUPs. And you also find intrinsically disordered regions. So here you're talking about proteins, here you're talking about regions or IDRs. But basically it's just referring to the same. Shown here in this publication of 2014, I like this figure, where you have a fully structured protein and you would have a fully disordered unstructured protein. So these are, let's say, the two extremes, and then you have proteins that are, let's say, in between, that are largely unstructured or largely structured, or let's say half-half is also possible. So different orders of structure unstructure, let's say. And then this was actually, if they looked in the ensemble release 63, they actually had a look at the protein coding genes in the human genome and at the different amounts of disorder, and then these are actually the results here. So 0 to 10%, so most of them are actually structured, but then you see still a lot of them, 3,000 between 10 and 20% and so on, and even a few then are fully unstructured. Now, how do you have to look at this? So this is the basic ID, the lock and the key that I referred to this evening. So ligand and receptor, a certain stable state, structure, and they can interact. What are other options? You can have what we call fold as you bind. And the gene regulatory protein CREB is one such example. That it uses a lock to mold itself into the shape of what we call the key. So it's not really structured beforehand, but then it can actually, when it interacts, when it binds, then it folds into a stable structure and it can interact. So fold as it binds the protein. And another example is shape shifting. So SIC1, signaling protein, is such an example. So it actually remains in a disordered state, and then you actually have these certain sites, 6 phosphate groups, and they actually can bind a certain site in turn. So you see that here the different ones, they can actually bind in turn this certain position. So what we call shape shifting. And there are structures also in PDB available where then you would find regions that are missing. And I already told you, if you would look into the PDB database and you look onto the sequence, that sometimes you see that part of it are missing. So in your protein sequence, it could be that in Pymol you will see these amino acids in gray, and you will find no coordinates, and you will not see them in the structure. One such region could be unstructured, and that is why you cannot see it in your Pymol. So for example, this is already quite some time ago, a Nature paper of 1995. In this case, signaling protein calcidurin, where you have a very large disordered region of 95 residues. Another example are the SH3 domains. And several proteins you can have these kind of domains. And then this is the one that I referred to earlier, the TP53 or P53, the tumor suppressor protein. So it has a stable region in the center and then lots of other regions in the beginning and at the end of the protein, and is unstructured and able to bind actually more than 100 signaling partners. So this is why this protein can bind also such many different partners. And then this is actually from the string database, how you can see the interactivity, YP53 with even a very high confidence level. So P53 is here in the middle. All of these proteins can actually interact with P53. So it's quite amazing. And this is actually one of the reasons why. Now, how can you find out that a protein is, maybe I will just turn on the lights. How can you find out that a protein is actually unstructured or partially unstructured or has unstructured regions? There are again some tools that you can use to do this, depending again on the algorithm. So sequence-based tools are Ponder and Disopret. B-factor threshold-based are Globplot and Dissemble. So remember the B-factor, last column of your PDB file, determining the position or is telling you something about the position where your atom is exactly, how reliable that is, the B-factor. Other subtle differences in predicting factors and tools that you can use are Run, Wiggle, and I will show you in a minute an example. These are the websites where you can find them. So maybe I will try Globplot and Dissemble. There is also a database of disordered proteins where you can find information. So Disprot is one database and another database is D2P2. So not R2D2 but D2P2. So I think that's the rest, and I think P53 is a nice example to illustrate that. So how will I do it? Gene NCBI, you know that database. This is also something you could get on the exam. So I go to the gene database, I want TP53, I add gene, remember, as a keyword, enter, I could specify and Homo sapiens. But Homo sapiens is always the first one. Then I click here. Then we prefer reference sequences. So on the right here, NCBI reference sequences. So you know messenger RNA sequence, protein sequence. I click on the protein sequence. 393 amino acids. I ask for the FASTA sequence. I copy the FASTA sequence and then I just type Dissemble, for example. This is one such tool, and another one, Globplot is another one. Just paste the sequence, and then what I do is here Dissemble protein, Globplot now, and this is why you see PostScript file. If I download it, you need such tool to have a look at the plots. So I already installed this tool. It wants to open. Yeah. So then it's like this, and with this, the EPSVS viewer can actually rotate it here. And then you might see, in this case, it's not that obvious, because you would expect this to be more stable, but I think the other one is probably more intuitive to interpret. So let's have a look at that one. Yes. So this is the result of the second tool, and then I take it here. That's easier to see, I think. So you see here the green region is what we call globular domain, so actually a structured region. The blue regions are disordered regions. So for P53 you see here from 150 until almost 325 this region is stable. And then here you have an unstructured region, so here a bit, and also here. What if I want to know which domains are present in this protein? Which tool can I use? CD search, conserved domain search. In the open book exam you can look at the course. So submit the sequence. And then you see here from 100 to just before the 300, I actually have the largest domain. So this is the structured part. And then, for example, this here, you see, before the 50 until 100, there doesn't seem to be any domain, known domain, right? It's actually this region. So you see that's unstructured. That's why there's no domain that you can find. There's no stable structure there. Same from the last part. If you look here, just after here, and this is actually a region I think that can form in a tetramer. But it may also may be unstructured, and once it forms a tetramer, then it might be structured. So, but again here, no information, and then I think you will also see here then, for example, the first part after the domain here is again unstructured, and here you also find such region. So this, in most cases, corresponds well, if you look at the conserved domain search, corresponds well with this. And this is actually, if you think how these proteins look like, we'll go to my presentation again. If, for example, you would have a protein built like this, with a CD strand, you have domain A, domain B. In most cases, these are folding, structured, and this here is just a linker region connecting the two domains. This is most cases then not that stable, that structure, unstructured. And then if you would check these tools again, you would see that this region is actually unstructured. And that is in most cases, if you want to do homology modeling, you will be able to find a template here that aligns and make a model. You will be able to make a model of this region of the protein, but for this will be impossible if you do homology modeling, because you will not find structure information. You won't find any targets in PDB. So in most cases, you can find from different domains and build models, but not for the, let's say, beginning, ends, and maybe between the domains, because often those regions are actually just unstructured, disordered regions. Okay, and I think that's it for all the chapters. Questions for the theory of today? So then, I will briefly go over the remaining workshops. So for the workshops, the first 3 are the most important ones. Why? Because there you have 10 examples, 10 nice examples, each time maybe a bit more challenging for you to try the molecular visualization. So it's important that you try those 10 exercises, that you're familiar with the commands, and that should make it easy for you to be able to do the 2 visualizations, one of a general structure, an overview, and one of a detailed structure on the exam. If you can make those 10 exercises, should be doable for the ones on the exam. It's similar level. Okay. And then the other 3 workshops.
You have to provide only the visualization or also the commands?
No, you don't need the commands, because there are always different solutions. So I will not look at how you did it. I will only look at the results. Maybe to illustrate, I can just show you the example exam. So the practical open book part. So the exam will be the same. A few questions on databases and tools is the first part of the practical. Then a general structure could be something like this. And then I will give points for, do I see the cartoon representation? Do I see maybe surface or sticks for a ligand, for example? Do you have a similar color scheme? And then for generating a figure. So what you need to always provide is a PyMol session file, so save it in a session file, and you have to render a figure. So the ray command doesn't have to be big, 800, 600 is okay. Ray command, and also upload the PNG. If I would have a problem that I cannot open your session file, at least I can give you score by just looking at the PNG. And also after the exam, when you submit, you come to me and we have a look at the view of the two solutions. So this could be one. So you also get, of course, the PDB code. So you just start fetch and then try to visualize it. And then the second part is then often a detailed view. So the most difficult part is not to have a look at a certain region. Can I zoom in to have the less or more the same view? And then of course there's always a part where you have surface or cartoon in combination then with the detailed interaction. Labels you do not have to provide. So no labeling, so you don't have to look at the labels. The labels are useful for you to help you to know which residues you have to put in stick, of course. That's why they are useful in the example that I will show you, but you do not have to provide the labels on the exam. What is Molecular Visualization Workshop 4 about? So there's also a small movie telling you that in more detail. You can actually also use Python scripts. Python scripts is actually your first course next year, scripting, but it's just a brief introduction. Just have a look at it. But I will not ask this, of course. Why is this interesting? It can help you. For example, you can, if you go to File, you can edit the startup file, which is the PyMol-RC file. You could, for example, add, because now I think it's white background in default when you start a program. It used to be black background. You can actually change all of these settings. If you want a gray background or another, you could just say BG color white, and it is by default always white background, or you can change it gray or some other color. Also, you can choose the 3-letter residue names. So it used to be 3-letter codes. Lis, cis, arg, and so on. Now I think the default is also 1-letter code. This is also something you can choose and set. So all of these settings, you can set that in your startup file. So that's one setting you can do. And then 2 examples in this workshop is you can just write a small script with just the same commands. So for example, hide all, show cartoon, you remember these commands? You can just use it cmd.hide, cmd.color, cmd.create, and then the options which you provide on the command line also here. So in this case, by default, I create an A chain, color them, B chain, color them, C chain, and so on. And then in the movie, you will see that if I just start fetch 1 BBB, run this script, all the coloring, all the settings are automatically done by the script. And I had students during the day, because it's always the same by default, you could say, split all my different chains in different layers. So you could just run prepare.py, create an A chain, create a B chain, C chain, and you will automatically have all your different chains in a different layer in PyMol. And else you will have to type 4 commands or more commands. So that's something that is possible to do with the script. On the PyMol wiki there are quite impressive scripts. One that I like is to find a sequence. Sometimes you're looking for a certain motif, 3-4 amino acids. But if you have a protein of 1000 amino acids and then you are scrolling, where is it? Do I find the motif? There's also a simple script that you can just search for it and it will highlight it in your structure, the motif. So that's, I think, also very useful. So you can download that script here, or I think it's also on the learning environment. And you have to make sure that, of course, the script is in your current directory. So in this case, when you start PyMol, that should normally be your home directory. Well, just check, because normally I use this in Linux. So it is the same pwd to see your current directory. Yes. So I'm now in my C Python 27 folder. So I would store this script in C Python 27 folder. Then I could just type run and the name of the script. then it will work. So in this case, the script here is what I call find sequence of Python. If I type run find sequence, then it works. And then I can sort by using the function find sequence, the name of the sequence I'm looking for, comma, in which object it has to look. So this could be a chain, but also your PDB codes. And then how it should save the result. So found will then be a new object on the right side with the name of the selection that will be created. So by just then using this command in your structure, you could find this motif and have it selected or saved in found. So that's often a very quick way of looking for a certain motif in your structure. And else it might take you some time to look exactly where it is, unless you know, of course, the exact position. But that's not always the case. And this is then, in this case, very useful. There are more scripts available. For example, one application that I had to use it for is I wanted to know the surface, how the interactions work, electrostatic interactions. And I think there's also a script there that you can just download and it actually colors your surface of your protein and show the electrostatic surfaces in red and blue. I think there's a script that you can just run to do that for you. So there are other applications. If you would ever have to do such thing, first thing is look in the script library on the Python wiki, and probably someone else already had to do the same thing and wrote a script and uploaded it there, and then you can just reuse it. So I won't ask you to write a script or run a script on the exam, to be clear. Just that you know that it's possible and it's quite nice, it can be quite useful. So that's workshop 4. And then workshop 5 is actually about the databases and just some examples. For example, I could ask you on the exam certain molecules or a family to look for certain sequences or tell me what is the official symbol. Then you have to look in NCBI Gene or in the HGC database. That you know the difference between protein RefSeq, messenger RNA RefSeq, where you can find that information. So these are such examples. Then this again here, domain architecture is again the CD search tool that you can use. Caspases, then you look in the PDB database, you could sort them then and have a look. Do I have caspase 1, do I have caspase 2, 3, and so on. Then this is just looking at the PDB website, how many chains there are, chain A, B, and so on. What is the resolution in angstrom, what is the R value, the R free value. You can just by looking at the homepage of the structure on PDB, you can answer these questions. Make a Ramachandran plot. You can have a look at PDB sum or at the Rampage tool. The Z-score, you have the Prosa web server that you can try for that. Structural alignment, one was the matrix alignment tool that you can use for that. Interaction partners is just knowing the databases, so the string database, remember I also showed you the P53 network is where you can find interaction information, but also on the NCBI Gene page and some other ones. And then this, of course, the docking experiment is something you will not have to do, but you can have a look and try it if you want. For this, there's again no, let's say, model solution, but one possible solution I provided you with a file where there's some screenshots. The text I think is still in Dutch. I need to translate that. But I think it can be nice if you don't really know how to do it. It can help you. For example, here for protocadherin 8, it tells you the query, how to look for it. Then here, how you have to search for caspases belonging to all mammals. So this combination. So then these are the answers actually. Maybe another one. For the CD search tool, I think you just have to change the threshold to find zinc finger number 7. Here. So the first result, you only get, I think, 6 zinc fingers, and then there you get an extra one. For the caspases, how you can find different ones. So this then would be the answer. For this PDB code, you type it in, then you find resolution, R value, R3 here. And here then in the macromolecule section on the PDB website, you find the different chains, the molecules, and then here normally you also find the genes, or if they are mutated or not. The result of the Ramachandran plot, the Z-score on the Prosa web tool, and then this is then the structural alignment in the mattress tool, how it looks like. So then this would be the result in that case. Okay, so just have a look at these exercises and maybe just try to reproduce the results, that at least for these tools you know how that works, where you can find the information. On the open book exam, of course, you can look at this, but if you have to look everything, then you might lose too much time, of course. So if you quickly already know, CD search is the tool to look for domains, for example, that can save some time already. Okay. And then I think there is one more, last workshop. And that's the structure prediction. So I'll just tell you about this, but this is something you don't have to do on the exam. But I think it's nice to see how you can do it. So how does the Pro server works? It actually, so you upload 2 files, one of your ligand, one of your receptor, and it starts actually by rotating your ligand for 70,000 rotations. And then for each, it translates in different x, y, z relative to the receptor. So it looks how it can fit or make contact with your receptor. And then of those 70,000 different rotations, it chooses 1,000 combinations that have the lowest score. Okay? So it looks how can it make contact, calculate score, and of the 70,000, the 1,000 rotation translations with the lowest score, it keeps those. That's the first step. And then the next step is it does a clustering based on the distance, root mean square deviation radius, 9 angstrom, of the alpha carbon atoms, and it tries to find if they are on the same location. So that's how it identifies what we call a clustered center. And then you get a result. So of the 1,000 you get, based on a cluster, you get a number of clusters as a result. How do you have to choose what is the most likely interaction? You have to look at the cluster size. So not at the score, but at the cluster size. So one example here was for a nanobody. In this case, what we did is open this structure in Pymol. We separated the 2 structures, so one file for the receptor, one file for the nanobody. We uploaded those on the Pro server and waited until we got the results. And then you will see of the 1,000 that were filtered out, the largest cluster here, cluster 0, is actually the first one, had 378 members. Which means of the 1,000, even more than 1/3 was actually at the same location, within the distance root mean square deviation of 9 angstrom. And then you had a number of other clusters. And then this was the result. If we would look at the results of cluster 0, the gray one is the receptor in this case. This is the ligand. The actual one in the experiment, the structure, is the green one. The prediction by Pro, cluster 0, is this one. I think pretty good result. Quite amazing, I think, that you can predict that. Of course, it seems to be, if you would have your templates that you use have a good resolution, that it will also improve the results. So another example to show you, this PDB structure actually had, let's say, bad resolution. Same experiment, separate receptor and ligand, run it on Pro server and have a look at the results. Here it is already more difficult. You see, the cluster sizes seem to be the same. So let's say in the range between 30 and 130, something like that. Also the scores quite similar. If we would then have a look at the top, they are actually on two positions. So you see here, this is again the receptor, the green one is the nanobody, this is cluster 0 and 1. They are actually at the same location. Cluster 4 is actually the correct one. But still, among the top results, the correct one was present. Only it was now not one of these, but here. And it seems to be, we already did some other experiments, that in most cases you find 2, maybe 3 positions here, and often the correct one is among those. So several clusters, and still, it's just because it rotates and the alpha carbon atoms, the distance is a bit larger, it is another center, a cluster center, but still the location on the interaction on the surface is similar. In this case for cluster 0 and cluster 1. So I think still a nice result that you could then tell, if you would of course not know the green one, you could tell, I think that the interaction is either here at the bottom or on this side of your receptor. And you only have 2 spots. Maybe then you can do an experiment or have a look at conservation or mutants and try to get a clue which one you might be able to exclude, for example. So I think you can have some nice results if you prepare your docking experiment and try to look at the results. You can draw some conclusions. And it goes quite fast. If you upload these 2 PDB files, sometimes within the hour or in a couple of hours you already get the result. So that's also quite fast. And then the last part is how you can make a homology model with modeler. For that you need 2 files. You need a Python script, a small one actually, it's always the same. And then an alignment file. The alignment file is the one you have to make for yourself. So you start with your initial alignment to identify your templates. And then you just improve your alignment, for example, based on the structure alignment. And then you have this format. And it's good to have, if there are several templates, to include them. The more structural information, the better, because it could be in the first region, one template might be more similar and provides better information, while for another part of your protein you're interested in, that another template is better to be using the information to make the model. So that's why often I use 3, 4, sometimes even 5 templates. And then in your script here you tell which alignment file to use, which known templates, so these are just the PDB codes corresponding what you provide here. And then how many models, so starting model 1, 3, ending model, which means I would make 3 models. Most cases I only make like 10, 20 models. And then I compare them and evaluate them. So this was one thing that I did, make a model for a nanobody. And then this was, I think, what I got as a result. This was done by somebody else, which you actually see, if you get a result like this, is actually a bad result. So if you would do modeler, automol, you would get a result like this. I think you already can see that there are lots of amino acids in disordered regions, so that this is a bad model. And then, so this is auto-modeling. Could be, I think it was with Swiss model. So fully automated. This was done with modeler with a couple of templates, and then you see that at least, of course it's still a prediction, that the structure and positions seem to be better and the model should be more reliable. With here a score of 90% in favored regions, which is, I think, nice for a homology model. Yeah, and then this is more detail about an example. So if you would want to try the homology modeling, there are two files. So RIP kinase 4 model alignment file and a Python file, and then you could install the modeler, run that, and then you should be able to do the modeling because you already have the two files required to do the modeling. So these are available here if you want to try it. Structure prediction, these two files. So install modeler, download these two files, run this script using that alignment file, and then normally you should get PDB files. And you can open those in PyMol. Why do I provide this? If in the future you would want to do homology modeling yourself, you can actually start from these files and then just change the script, change the alignment file, and then make models of your own. But then at least you have two template files to try out. Okay, that's, I think, all I have to tell you. So any questions or problems or about the exam? It's already next week, so you don't have 2 weeks. That's why it's a bit less. I try to put more into the beginning. So I think definitely study very well the first 3 chapters. Make sure you try the 10 molecular visualization. Have a look at the tools, and then I think that's already a good start to prepare you for the exam, and then try to have a look at the rest of the theory. Not into too much detail, have a look at the example exam, so it's not always in too much detail. I don't ask names or dates of people, for example, so please don't study those details. So it's, yeah, I think that should give you an idea how to study and prepare for the exam. Okay? And then I will see you next week.
Isso.